Hi, everyone. We'll get started in just 1 minute. Okay, I can see we have a good number of people logged on. So just want to welcome everyone to the Division of Rheumatology Grand Rounds. Today we have Dr. Mark Wener with us, and he is presenting as part of our UW Research Pioneers lecture series. Just a couple reminders before we get started. Dr. Wener will save about 10 to 15 minutes for questions at the end of his lecture. And so please try to use the Q&A box at the bottom of your screen to type your questions in, as opposed to the chat box. And that way all of our questions will be centralized into one place. And now I will pass things off to Dr. Mark Wener. Thank you.
Thank you, Laura. And again, I appreciate having the opportunity to speak about my mentor and long-term colleague and friend, Dr. Mart Manick, who, one of the ways that I met him early on in my career was going to an ACR meeting before I was in the division, and at a plenary session on immune complexes, he was introduced as Dr. Immune Complex. So that's the title of my talk today about Dr. Manick. Here's a picture of him. As long as I knew him, he had that white hair and really almost military bearing, and eyes that when he was in a good mood were playful and fun. And if you were presenting data that didn't seem right, they would look right through you. And as fellows and colleagues, we knew Mart in both ways. He had a strong identity for data and a faith in data and wanted it to be done right. Here are some key academic dates in his life. He graduated from Western Reserve University in Cleveland before it was Case Western, did internship and residency at MGH, was a fellow and guest investigator at the Rockefeller with Henry Kunkel. I'll get back to that later. Then he went to the NIH for a couple of years, was recruited back to the Rockefeller University as an assistant professor, and then came to the UW in 1967 and joined the emeritus faculty after his formal retirement in 2001. And then passed away 2 years ago. Look at the date, by the way, of course, this is almost would have been his birthday. But milestones at the UW, my gosh, 34 years as division director. He trained over 100 fellows. He came here as, still not a senior, well, not a very senior faculty member, age 35 in 1967. I note that his first publication from the UW took a few years, and I think that reflects the fact that he was building the division not quite from the ground up, but nearly so, and in his image. And it took several years to get going and get started. He was then promoted to full professorship in '73, and the Mart, Manick, Lucille Henderson Endowed Professorship in Rheumatology was established in 2004. Important for understanding his history, and I think partly perhaps personality, as he came to the United States as a refugee of World War II from Estonia. And again, thinking about Estonia in 1932 with Germany to the west and Russia to the right, and the other Baltics here all kind of compressed between the two, of course, and Poland here. And news of the day and of his childhood and youth. Worthwhile remembering these perspectives as our own country is dealing with challenging times and trying to think about what Mart went through as a youth. But he, and again, worthwhile, I think, also contemplating and thinking about and reflecting the way the US has welcomed refugees and how important they are to our country. Mart was married to his wife, Zita, for 42 years, and they were gracious hosts. They traveled the world, both while he was working and in retirement. He had 2 kids and a stepdaughter. By the way, Henry, I think named in part after Henry Kunkel, his mentor at the Rockefeller. Couple thoughts about Mart and his personality, he was an avid skier. And the story was that he taught himself. And the image that Zita would talk about is that when he went up on the slopes for the first couple times, he brought his textbook of skiing with him and would read about it and then try to ski and then read about it and then do some skiing. And you could see this bookish man and young man learning to ski, and both the independence that he expressed and the way that he sought knowledge and gathered knowledge and had faith in the written word and in writing. While in the Seattle area, Pacific Northwest, he skied, hiked, kayaked, was active as an exerciser in health clubs and others and other ways personally, but he did not watch team sports. And I think one of his elements of pride is that in his many years here, he was never in Husky Stadium. Even though he at least once was tempted. And I can say at lunchtime, he used to lead the division fellows and faculty to go to lunch together at the South Campus Center or kind of right around. Then we would often go on walks and usually walk down along the cut to fisheries or oceanography and look at the boats and the deep ocean biopsy aliquots, et cetera. But one year in 1990, the pseudo Olympics friendship games were being held in Seattle at Husky Stadium. And he decided we should take a walk in that direction that day so we could peek in, but he never went inside. Let me talk about Henry Kunkel and the Rockefeller. Mart trained with Henry, who and had lifelong friends that stayed all of his life from that time. And at that time, there was an amazing and talented group of young investigators, many of who became leaders in rheumatology and immunology, other areas. Many were rheumatology division directors, et cetera. It was a vibrant community of budding academicians. By modern lens, not the diversity we would appreciate and welcome now, but I think a real crucible and place to really be developing scientific ideas. And they just grew under Henry Kunkel's leadership, and Mart really inherited and passed along that tradition. So while Mart was there, he was investigating many proteins, including myeloma proteins, became an immunochemist. Along the way, he had access to a variety of serum and plasma specimens that came from patient care at the Rockefeller, and including, in fact, the story goes, he had some of the serum from the original SM and participated in patient care. So just to break up the Zoom for a minute, how is SM antibody system named? Is it associated with smooth muscle antibodies? Was it originally identified by Dr. Smith, identified by a patient Smith, or is it abbreviation for a subcellular particle? And if anybody's responding, I can't see it, so don't bother, but think about the answers to these questions. Wait, maybe I can see. I can't see. Anyway, it's not important. I think everybody knows the answer. Identified in a patient named Smith. But the important part is that while at the Rockefeller, Mart helped care for Mrs. Smith, after whom the SM antibody was named. Partly with this continued connection and affiliation and feeling for Dr. Kunkel and the Rock, the Henry Kunkel Society, which was formed initially by former trainees from Dr. Kunkel's lab, that organization was started at a meeting in Seattle when the ACR was in Seattle and was hosted by Mart and Zita. And that society has continued to grow. And in fact, Dr. Elkan is a member and former president of the Henry Kunkel Society, which continues to promote high scientific rigor in rheumatology research. Mart had a number of national honors. He was a distinguished investigator of the ACR. He won the Howley Prize from the Arthritis Foundation. Again, by way of background, the ACR, American College of Rheumatology, of course, is the rheumatologist professional organization in the United States. But for many years, it was actually part of the Arthritis Foundation. And so the AF and the ACR were really closely linked. And much more involvement of the AF in research, because that was the rheumatology organization was part of the AF. Mart became a master of the ACR, and among his honors, I would say, and recognition is, in a sense, he literally wrote the book. And so this is Harrison's Textbook of Medicine, and from the 7th edition in 1974 until the 10th edition in 1983, virtually all of the rheumatology chapters were authored by either Dr. Manick or Dr. Gilliland, in that order, or Gilliland and Manick. And there were a couple exceptions, I think, where Dr. Gilliland or Dr. Manick wrote separately, but Gilliland and Manick were rheumatology for generations, if you will, or at least hosts of medical students who learned from Harrison's. And also the main rheumatology textbook of the time from the United States was McCarty's book, and Mart wrote the chapters on immune complexes and on rheumatoid factors for many years and many editions. And I wanted to spend most of the time talking about his research and research interests. I want to present some actual data. And I think actually in honor of Dr. Manick and what he valued, I think he would appreciate that we're going to look at some data from lab experiments that he generated with fellows and trainees along the way. Mostly this is going to be an overview, but we're going to have some primary data in Mart's honor. So thinking about lupus nephritis, it's an immune complex disease, at least in part. We know it's complicated. Here's immunofluorescence staining of IgG and C1q in the kidney of a renal biopsy from a patient with lupus. And among the questions Mart was asking was, how did the immune complexes get there, and what controls them while they're there? Could you get them out and maybe help patients? And specifically, what are some biochemical features of the antibody and the proteins that are antigens that can influence this process? And so many of his doctor immune complex kind of tradition was based on serum sickness and previous experiments with model immune complexes. The main experimental immune complex system that Mart used was human serum albumin, HSA, as the antigen, and rabbit antibodies to anti-HSA as the antibody. And the era, they would make their own antibodies, so they would can purchase albumin, but then have to purify it and then inject it into rabbits with Freund's adjuvant and make their own rabbit antiserum. There was no buying reagents off the shelf, certainly in the early days of Mart's career. And among the important observations were that the size of the immune complex and the electrical charge on the immune complex proteins influences their removal from the blood and influences their deposition and persistence in the kidney, the main target organ, or certainly important target organ of lupus nephritis. And really thinking about the implications of the pathogenesis of lupus and other immune complex disease. So some methodologic notes, again, in honor of Mart. We're going to go to the method section of some of his papers, but partly this is way of background. So the size of proteins were in his era were determined largely by analytical ultracentrifugation using sucrose density gradients measured in Svedberg units. And we still sometimes use the S nomenclature, normal IgG, which of course is around 150,000 KD, but in analytical ultracentrifuge sediments at about 6.6 to 7S, normal IgM is sediments at 19S. And he spent a lot of time looking at sort of what might be called intermediate size immune complexes, AG2AB1 or AG1AB2. And this became kind of his standardized immune complex reagent. He used others as well. And worthwhile thinking about precipitin curve kind of in the context of immune complexes. So if one has a constant amount of antibody and add an increasing amount of analyte or antigen on the y-axis, the amount of precipitate forms initially in a zone of antibody excess because there's more antibody present, and then there's another zone of antigen excess and a zone of equivalence. And what Mart did would take the soluble immune complexes, the antigen antibody complexes formed in antigen excess. And these had a range of sizes, but he would throw away, if you will, spin down the precipitate, but look at the soluble complexes and inject them into mice. And introduced and really spread the idea of an immune complex lattice, that different immune complexes of different sizes could be thought of a little bit like a crystal structure in a way, like a lattice. So the small immune complexes shown here in the top left, these intermediates in the top right, where each Y, of course, is an antibody, and each circle or oval is intended to represent an antigen. And then on the bottom, these large extended lattices that would be very, very large. And he could separate these by sucrose density gradient ultracentrifugation. The bottom of the tube is on the left. The top of the tube that was spun is on the right. And the 6.6S or monomer normal numeric IgG would be separated from the very large ones or from the ones that are in the middle. And he could pool these and inject them and see what happens. And so this was actually the first publication from his time at the UW. And notable, you can see Dr. Arendt, who we spoke about at this series a few months ago, and Dr. Gilliland, again, longtime professor and one of the leaders in the division, were all co-authors. And this is about elimination of soluble immune complexes that had been injected into rabbits. And this is an example of some of the experiments that he would have done. So remember the human serum albumin and anti-human serum albumin that's radio labeled. They can inject them into mice, time on the x-axis, the amount of remaining radioactivity on the y-axis, and look at the clearance from the circulation. And what they found is that normal monomeric IgG cleared with this kind of pattern, and these small and intermediate immune complexes actually persisted in the circulation for a while, whereas the largest immune complexes just basically inject them and they're gone very, very quickly from the rabbit circulation. And then the question is, okay, they disappear, but where do they go? And in this case, Bill Arendt, who was Mart's first fellow at the UW and went on, of course, to a distinguished career, he was the first author of this paper that addressed the question, where are these immune complexes going? And again, here's the data. I'm not going to go through in detail on all the things that they did to investigate it, but what they found was that the initial immune complexes were cleared from the liver, in the liver very rapidly. That's where the radioactivity went. And overall, about 40% were gone in 40 minutes. But the largest immune complexes, those things in the bottom of the ultracentrifuge tube, cleared even faster, about 90%. And of interest, and I think maybe a little bit of a surprise, it didn't depend on complement activation, even though it did depend on the FC region of IgG. And by then, it was recognized that complement activation was dependent on the FC region. And what they used, as shown in this table, is they used cobra venom, which could remove the complement chemically from the rabbit that was injected, or used aggregated IgG, which consumed all the complements, so there wasn't any complement left. And it really didn't influence where the uptake in the liver of these immune complexes. But if you altered the FC region, and they did this chemically by a technique called reduction and alkylation, very different finding, and they didn't show up in the liver nearly at the rate. So among the early conclusions was that there are some important physical chemical issues about immune complexes. Large immune complexes were known to activate complement and had been thought likely, therefore, to be most pathogenic. But those largest complexes are rapidly cleared by the Kupffer cells in the liver, the essentially mononuclear phagocytes in the liver, and therefore maybe aren't responsible for end organ disease. And the process does depend on the intact FC region, but doesn't require complement. But his thinking then was, well, the small and intermediate complexes that persist in the circulation may have more potential for causing disease. But it also pointed out a key role for macrophages, for example, the Kupffer cells in the liver, in causing immune complex clearance. And so Bill Arendt, still as a fellow working with Mart, continued along those lines of investigation. And again, done in a very quantitative way, looking at the number and the affinity of binding sites on the macrophages, looking at how they immune complexes adhere to soluble immune complexes, the same HSA, anti-HSA. And actually, Bill Arend, as I said, went on, but his initial description of the interleukin-1 inhibitor, published in 1985 in Journal of Immunology, which we now know as the IL-1 receptor antagonist, was in part based on the effects of these HSA, anti-HSA immune complexes that he started to study in conjunction with Mart and their effect on human monocytes. So thinking about monocytes, and again, this is getting the those of us who are studying for boards to think about 2020 and even 2021 knowledge, thinking about Fc gamma receptors on phagocytic cells, which we know are at least in part responsible for the clearance of immune complexes. So some questions to think about for Fc gamma receptors on phagocytic cells. Do they have a role in immune complex clearance, as Mart and Bill Arend really worked on, but no immunoregulatory role? Or do they also activate cells when immune complexes bind? Or can they inhibit macrophages? Or can they both activate or inhibit? What do they do? Well, they do both. And most Fc gamma receptors activate, and there's an immunoreceptor tyrosine base activating motif, so-called ITAM. But the Fc gamma R2b is inhibitory. And so, I think we're thinking about immune complexes in different ways. In this era, thinking about their immunoregulatory role as well as their role in immune complexes clearance. But back to Mart's studies, and now working with Al Hackenstadt, who was a PhD student and then became a fellow and then later on in faculty. And what they found is that if you injected a lot of immune complexes, and now working with mice because they could do lots of experiments on lots of animals, as opposed to the rabbits they were working on before, what they called the reticuloendothelial system, the era's term for the mononuclear phagocyte system, those macrophages could be overwhelmed and saturated. And here's, again, some data looking at different amounts of immune complexes ranging from 0.1 milligrams to 5 milligrams, and looking at the clearance curves from the circulation and the slopes that were different as derived from these experimental conditions. And the clearance was relatively rapid with small amounts of immune complexes, got a little slower as the numbers got up. It became very slow when you overwhelmed the mononuclear phagocyte system with immune complexes. And this shows the clearance with the amount of added complexes, and these are now these large immune complexes, remember, that before were cleared very rapidly. If you added more and more of them and looked at the clearance velocity, a lot of it was cleared when you didn't add very much, but after a while the system became saturated and clearance essentially stopped. And this overwhelmed the system. And I would say this is overwhelming, is looking at this table of data, which I'm not going to walk through, but overwhelming both about providing the data about overwhelming the mononuclear phagocyte system, but the overwhelming experiments that were being done. Look at the number of mice that were done in these experiments. And I think the notion was you do the experiment, and then you think about what you might have done wrong with the experiment, and then you do it again and you do it again until you got lots of data and lots of numbers to generate reasonably tight elements of precision. So lots of work and very careful work, which was a hallmark of the way Dr. Manick did his experiments. So we've talked about importance of size in how and immune complexes and how they get cleared from the circulation. But then he studied how important is that for what deposits in the glomerulus if you could overwhelm and saturate the mononuclear phagocyte system. And this is a series of experiments done in the early to mid 80s about the disappearance kinetics, and this is about small lattice complexes. He looked at small, medium, and large, and a number of different factors. And then starting in the kind of mid-80s, also looked at another physical chemical property of immune complexes, which is their electrical charge. You could modify the proteins in either the antigen or the antibody, adding cations, for example, and look at their effect on the deposition, in this case, and persistence within the glomerulus. And again, not too much numbers to look at, but just looking at the numbers of pluses. If you had these cationized antibodies compared with the antibodies alone, compared with the native antibody and native antigen antibody complexes, this is what's shown here, you could show that in compared with the normal antibodies, the cationized antibodies, even in small amounts, deposited in the kidneys. And the amount that they deposited was even more than immune complexes. But whereas the antigen antibody complexes with normal antibodies ended up getting cleared out of the circulation, out of the kidney after a couple of weeks, when you had these cationized, electrically charged antibodies, they just stuck around for a long time, particularly when there was a fair bit that was added. So importance of both size and charge in influencing what goes to the kidneys in experimental systems. And also worthwhile thinking about where they go in the kidney. So this is some electron micrographs showing the ultrastructure of a glomerular basement membrane. And this is a normal glomerular basement membrane. Here's the capillary down below. Here's the urinary space up above, the foot processes of the epithelial cells, and here's the endothelial cells down here. And what you can see with high magnification is that the glomerular basement membrane actually has three regions. There's the so-called lamina rara interna, which is in the subendothelial region. There's the lamina rara externa in the subepithelial region beneath the epithelial cells of the kidney, and the lamina densa in the middle. And so there's some interesting sort of physiology, anatomy that goes on within the glomerular basement membrane. And in fact, if you want to know more about that, one of the investigations that was done was to take ferritin, which of course contains iron and therefore could be seen on an EM, and cationize it, make it electrically positively charged, and then inject them into experimental animals and see where they go in the kidneys. And you can see that the cationic ferritin decorates, sort of has these little dots along the lamina rara interna and externa. So these are the anionic sites within the glomerular basement membrane. And this kind of approach allows one to be thinking about what's the potential role of electrical charge on antigens and antibodies and what goes on. And so they use this kind of approach to look at cationic antibodies and cationic immune complexes to see where they go. And the cationic immune complexes by themselves would act like cationic ferritin. They would so-called decorate, if you will, deposit in small amounts, put these little dots on both lamina rara externa and interna. And that was actually also true of immune complexes. Some of the arrows go up, some of the arrows go down, both the subendothelial and subepithelial areas. At 12 hours, the antibodies alone are still there, and they do actually cluster a little bit. These are larger dots than they were initially. But when they're immune complexes, the clusters are getting bigger, some of them getting quite large, and they are mainly focused in the subendothelial region at the lamina rara interna. And if you look at even later time points, still a few dots if you inject the cationic antibody alone, but now there are these large and more extensive immune complexes present that have coalesced. Still a few dots on the subepithelial side, but these big immune complexes are assembling in the subendothelial region. And he further explored this idea of rearrangement of immune complexes by forming immune complexes that were now chemically structured so they couldn't rearrange. So these were modified antibodies. And this is a paper published with Larry Agadoa, who ended up leaving. He was a nephrologist in the division doing these experiments and went on to become the head of the U.S. Army's nephrology division. But these are experiments done to look what happens if you prevent immunofluorescence, if you prevent rearrangements. And I'm not going to go into the details, but these so-called cross-linked complexes prevented persistence and formation of these large electron-dense deposits in the glomerular basement membrane. This is some electron microscopy to demonstrate this. Here's a mesangial cell. Here's the subendothelial regions. And if you have these normal rearrangeable immune complexes, you get these dense deposits showing up in the subendothelial region and adjacent to the mesangium. And of the non-rearrangeable immune complexes, given the same dose, you don't form this. So rearrangement is important. So here's another kind of question to think about. What is the significance of subendothelial versus subepithelial immune deposits in lupus nephritis? Is it no difference? Is it that subepithelial are associated with class 4 lupus nephritis and subendo with class 5? Or is it the reverse? Or is one of them associated with tubular disease? And the answer is subepithelial deposits are part of class 5, membranous nephritis, and subendothelial associated with class 4. And actually, this is from kind of a little illustration of this from Kidney International's proposed revised classification of lupus nephritis. And here's more or less normal class 1 glomerulus. These are obviously pictures of or cartoons about glomerulus. Here's class 3 and 4, that is the proliferative forms of lupus nephritis. And shown in black here are the subendothelial deposits and also immune deposits in the mesangium, which is where they are found in patients with proliferative forms of lupus nephritis. In contrast, in membranous lupus nephritis, such as class 5, the immune deposits are in the subepithelial region, as shown as the black spots here. And this is the paper. I just took those cartoons directly from this paper. So subendothelial electron-dense deposits likely reflect immune complex disease that has rearranged and condensed these immune complexes. So the GBM at some level is actually a fluid structure and dynamic. And it's kind of interesting to think about. Over the years, we've collectively in medicine said things that look like they're permanent must be inactive. And we've mistakenly thought that about osteophytes. We've mistakenly thought about that for a while about chondrocytes. We've mistakenly had that thought about fat cells, adipocytes. And in a way, we didn't think that immune complexes in the kidney were dynamic. And I think part of what Mart led us to understand is that they can be. They are fluid, and immunochemistry can happen there. And so if that's the case, could you remove them? If you have antigen antibodies that are recently deposited before they get buried in layers of matrix, and he did the experiment that looks like the precipitin curve, where if you add more antigen, you go to antigen excess, you can potentially make the immune complexes that were larger, make them smaller, and potentially remove them. And he demonstrated that. Again, I'm not going to show that primary data. But subendothelial immune deposits then do have an important role in sorting out mechanism of disease and how we think about disease. They're part of proliferative lupus nephritis, class 3 and 4, as I mentioned. They're also seen really in various forms of membranoproliferative glomerular nephritis, such as associated with hepatitis C, virus infection, and the syndrome of mixed cryoglobulinemia. And I think one might say that mixed cryoglobulinemia is really the main clinical example, strongest clinical example of the role of circulating immune complexes and their pathogenic potential. He showed that the subendothelial deposits are caused by deposition mostly of preformed immune complexes. I may have time to develop this a little bit later, but not always, and often associated with deposits in the mesangial region because the renal macrophages in the mesangium essentially are involved with clearance. But they also form because of rearrangement and condensation and movement within the glomerular basement membrane. They're not just showing up there and then they're fixed. They show up there and they rearrange as a dynamic process. In contrast, subepithelial immune deposits associated with things like idiopathic membranous nephritis and class 5 membranous lupus nephritis are probably more often related not to circulating immune complexes that deposit in the kidney essentially by being filtered, but by planting an antigen or antibody first, and then followed by the antibody or the antigen that's the cognate immune reactant. Historically, by the way, UW was a very interesting place in the mid to late 80s because the major proponent of the notion that immune complexes are formed in situ within the tissue itself was Dr. William Kauser. Bill Kauser was the head of the nephrology division here at the UW, while Mart, of course, was the head of the rheumatology division. Mart was a circulating immune complex that have the central role, and we don't think there's very much importance for in situ formation, and Dr. Kauser had the exact opposite view, and they would debate it, and it was actually pretty interesting to see these really wealth of knowledge and wealth of perspective coming at it from different angles, and is often the case. Truth is a little bit of both, and so it turns out both mechanisms are important in human disease. But if the circulating immune complex hypothesis is really likely to be true, we ought to be able to wash out those immune complexes. And so important paper from Dr. Verrier-Jones in 1976, and the title is Plasmapheresis in the Management of Acute Systemic Lupus Erythematosus, ? All right, published in The Lancet. And so we and others with working with Mart said, well, if that's the case, can we use all this information that's been derived from rabbits and mice and look in humans to see what are the immune complexes in lupus patients? And what was done was to look at C1q binding. C1q, of course, is the first component of complement whose role in life in part is to bind immune complexes. And that was used as an assay to measure immune complexes in the circulation. In the late 70s to mid 80s, there was lots of interest in laboratory tests for measuring circulating immune complexes as a disease activity marker for lupus and related disorders. But we could use some of the same techniques of sucrose density gradients and apply this to human serum and measure where the C1q binding activity was. So the green arrow, this is 6 different patterns representing a number of lupus patients that we examined. The green arrow shows where the 6.6S or monomeric IgG migrates. And one can see that there's a range of immune complex, C1q binding. Some of it is all in the heavy region. Some of it is a mix, but a lot of patients had it mainly associated with monomeric IgG and normal serum. And this was further investigated by Shu Uwatoko, who was an investigator, a nephrologist from the University of Tokyo, who came to the lab and really with Mart definitively proved that the C1q binding IgG in lupus is mostly not immune complex, but anti-C1q, and it binds specifically to the collagen-like region of C1q. So here's the C1q molecule. As shown often like a bouquet of tulips. The globular head is the region where immune complexes bind and mediated through the FC region of the IgG that binds the immune complex. And the stalk of the bouquet of tulips is collagen-like region of C1q. And this is where anti-C1q binds. And you could demonstrate that by removing the globular head, by breaking up these autoantibodies, by a variety of other factors, and prove that most lupus C1q binding is not an immune complex, but anti-C1q. And in a way that was shocking. But further investigation says, yes, not only does it happen, but it's true primarily in lupus. These are controlled populations with Sjogren's and RA and normal serum. And it doesn't happen to all lupus patients, but primarily a subset and often associated with renal, with lupus nephritis. And in fact, if you look in the kidney, in the kidneys of patients who died with lupus nephritis, you could find autoantibodies in the kidneys, including anti-C1q. And so with further investigation that Mart did is you could in fact look at autoantibodies that were enriched in lupus kidney glomeruli. And I think everybody expected double-stranded DNA to be in the kidneys and to be the major cause of the immune complexes in the kidneys of lupus patients because of the long history of double-stranded DNA as an antigen and anti-double-strand DNA. But I think many people thought these other antigens were just kind of there as to be measured by antibodies and maybe didn't have a pathogenic role, and certainly not in lupus nephritis. But if you isolate the glomeruli from lupus, you can find multiple autoantibodies in these immune deposits. SM, SSA, now these are the antibodies that are present in the glomeruli: anti-double-stranded DNA, SM, SSA, SSB, chromatin, anti-MPO. There was evidence for histone antibodies present, although I put the question mark because histones and antihistones are so sticky that it's hard to be sure about. But the observation also was that anti-C1q tended to coexist with other autoantibodies and to be associated with these proliferative forms of lupus nephritis, which is also associated with subendothelial deposits. So I'll link them back. And so sort of here was the model that you have an immune complex that binds to the kidney. C1q comes along with its bouquet of tulip structure, binds to the Fc, but these need to coalesce and bind together. So along comes anti-C1q and leads to potential cross-linking of these so that they coalesce and also complement activation, so that anti-C1q has potentially a direct role, but really an amplification role in augmenting immune deposition and subendothelial deposits in the kidney. I would be remiss to say if I didn't talk about double-stranded DNA itself, and working with Woody Emlyn, another one of Mart's former fellows and then faculty at the U for a while, looked at DNA size and strandedness and looked at in vivo clearance in animal models of DNA, but I'm not going to go on to this. Just there are other areas of investigation that I'm not going to spend much time talking about. But work not from the UW, but in the field of medicine, there was a controlled trial of plasmapheresis therapy in severe lupus nephritis, and it was a bust. I don't have the data. The data basically, it's like overlapping lines compared with patients who didn't get plasmapheresis for lupus and those who did. There was no benefit. And if anything, just a little bit of fluctuation in statistics, the people who did plasmapheresis did a little worse. So the trial was stopped before it was officially scheduled to be finished because they said even if it doesn't make them worse, there isn't sufficient power in the remaining part of the study to show that it's going to be benefit. So end the trial early. So that actually also reflects that the interest in circulating immune complexes as measured by the number of publications in PubMed that I looked at that have this title peaked in the 80s and then sort of has declined. And there's still interest, but it doesn't have the same interest. In a way, I think it's ready to be resurrected potentially, but doesn't have the interest now. Immune complexes in general also peaked as a topic in PubMed, not circulating immune complexes, but immune complexes in general, peaked in 1983 also, and has maintained more of a steady level since then, and not the steep decline of circulating immune complexes. And part of this is work done from Dr. Elkan's lab and Christian and many others about different kinds of roles of immune complexes and how we think about it. This is still a cartoon of an immune complex centered view of lupus, but in addition to the work on tissue deposition and the role of complement in tissue damage, we now know about immune complexes and their role with apoptosis and netosis and potential activation of type 1 interferon signaling and much more role potentially as influencing macrophages and their potential role. So lots of continued interest in immune complexes, but now this is one portion, and there's an expanded portion of what's going on in thinking about immune complexes and their clinical role and their pathogenic role and scientific interest. So in kind of summarizing Mart's kind of interest in immune complex diseases, I think his experiments were often very detailed, quantitative, very careful, very precise. He was persistent. He would follow the idea. He was curious about these ideas. He had attention to lots of detail. He really set very high standards for himself and for those around him and asked probing questions. And he never wanted to go beyond the data. He did not like to publish hypotheses. He said, here's the data, let's explain it, and then we'll do more experiments if there's a hypothesis we want to test, but we're not going to go out on a limb, if you will. And I think that reflected his scientific personality and was one of the strengths of the kind of work that he did. I want to develop another aspect of his research, and I'm going to leave some time at the end, I hope, but I need to talk about rheumatoid factors and anti-antibodies and Mart's role in that. And this really goes back to his time in Henry Kunkel's lab, where the initial descriptions of idiotypes, that is unique antigenic determinants on immunoglobulins, which was really a shock, I think, when originally discovered in 1963 and 1965, led to a publication in Science at that time that basically said, you know, these antibodies, we kind of thought they were mostly the same. We know they recognize different antigens, but chemically, would they be so different? And what they did was they took myeloma proteins from patients and they injected them into rabbits, made rabbit antiserum, did octolony double diffusion, where the immunoprecipitation lines can show reactivities and cross-reactivities between different serum with different rabbit antiserum. And what they found is that there were a few antisera that reacted only to the myeloma protein from a single individual. And they kind of presciently said, you know what? The localization of individual specificity to the FAB fragment, which they demonstrated, is of considerable interest since the antibody combining site of antibody molecules is located on this fragment. And the fact that these myeloma proteins are apparently antigenically unique in this portion of the molecule suggests that there's a very great variety of grossly different structural forms in the FAB fragment of normal gamma globulin. Well, that was novel. And again, by background, heavy and light chains weren't clearly well understood. Kappa and lambda as antigenically different was just starting to get studied. So this was novel information. But continuing some work on antibodies, and by the way, what else is a rheumatoid factor but an antibody to IgG? Working with Dick Richard Pope, who was another one of Mart's early fellows, had a distinguished career as the division director at Northwestern for many years. They looked at the self-association of IgG rheumatoid factor. And again, sucrose density gradients. I'm not going to go into the details, but you are already experts. Here's IgG, here's IgM, etc. And what they found was that there are these intermediate immune complexes formed just by adding the isolated rheumatoid factor from a patient. And they proposed this model after a lot of work, that there's this so-called self-associating IgG rheumatoid factor, where the FAB of one IgG rheumatoid factor binds to the FC of the second one and vice versa. And this idea of this extended lattice of immune complexes formed by rheumatoid factor is in fact a rare cause of hyperviscosity syndrome. And led to a lot of interest in, or substantial interest at the time in studying rheumatoid factors. And by the way, this figure was on the cover of a rheumatology textbook in the 1980s. Not quite the Rolling Stone, but still got lots of recognition. Mart did a lot of other work with various fellows and faculty on rheumatoid factor and its connection with microbial FC binding proteins. So microbial FCs, microbial proteins in many instances bind to the FC portion of IgG, very similar location to where rheumatoid factor does. This is work led by Fran Nardella, who was a faculty member for a number of years, and Eric Sasso, then a fellow and later faculty. And in this little hypothesis picture, they said, okay, rheumatoid factor binds to the FC region, but so does staphylococcal protein A, and staphylococcal protein G, and HSV1 has a binding protein, and T15, and all these binding sites for these microbial proteins might look a little bit like, or they look like the binding area of rheumatoid factor. So they proposed actually that rheumatoid factor is an anti-idiotypic antibody. So let me walk through this. The microbial infection leads to production of microbial FC binding protein, which binds to the normal human IgG. Patients make antibodies to these foreign proteins. Then, as part of the normal immune response, patients make an anti-idiotypic antibody to the FC binding protein, and that is kind of essentially what a rheumatoid factor is, binding to the FC of IgG. This is part of a larger hypothesis, the Yerny hypothesis of the idiotypic networks with so-called internal image. But anyway, this was the direction that Mart's lab went. But of interest, they continued to study staphylococcal protein A, which was really well known to be an IgG binding protein, binding to the FC of IgG. But it also was found to bind to a small amount of IgM and IgA. And the answer is why. And when they further investigated this, they found that some variable regions, now in the V region of the molecule, bind staph protein A. And the variable region gene is actually a conserved sequence part of the V region family, the VH3 family, and that this is expressed in germline of immunoglobulins, the variable region. The germline, prior to rearrangement, has this intense potential for binding. And so they proposed that actually staphylococcal protein A is a little bit like a super antigen. It binds to the variable region of a IgG molecule, not through the usual FAB binding site, but in an area next to it on the side, if you will. And so a whole family of IgG molecules and IgM molecules and IgA molecules can bind to staph protein A this way because they have this germline encoded sequence that binds to that region. So I just wanted to present some of the range of interest that Mart had, but also tenacity. He really asked the same basic questions, I would say, for most of his career. He altered and refined lots of thinking about immune complex disease and its kinetics, quantitative aspects, thinking about the glomerular basement membrane as a dynamic structure for immune complex kinetics. And then really contributions to the UW that are really remarkable. You know, he set up the immunology research program in the rheumatology division and set it on its course. He provided stability of the program for many years. He attracted research and clinical faculty, trained literally over 100 fellows, and I feel myself fortunate to be among that group. And our program trained most of the rheumatologists in the region and many around the country, and including noted scientists and clinical leaders and investigators around the country. So I think we owe a lot to Mart. And again, I feel honored and a privilege to be able to tell you some of the things about him. So thank you for your attention.
So Dr. Wener, it looks like there is a question in the Q&A section. Do you see that?
Yes, thank you.
Mm-hmm.
So a good question. What is the origin of anti-C1q antibodies? And one might generalize to say, what's the origin of autoantibodies in general? And the short answer is, I don't know. One might continue along the line of Natalia's question, which is a good one, is could C1q become immunogenic in lupus patients? Are there neoepitopes that might be generated in various situations? We know that C1q binds many different structures, including a work from Keith and others about binding DNA. And is it possible that there are neoepitopes that are generated? I mean, I don't know the answer to the question, and the same way I don't know why most autoantibodies get formed, but is there a potential role because of immune complex formation? I think that's an interesting idea, Natalia, and certainly possible.
Okay, so there is just one other comment from Dr. Gardner. I don't know if you saw that, but he says, thanks, Mark, for a wonderful presentation about Mart.
Oh, thank you. And again, I know you also had the opportunity of being with him. So thank you.
And another question just popped up from Dr. Jernberg.
Yes, thank you, Elizabeth. Yes, how helpful is measuring C1q binding in clinical practice? You know, another good question. We used to offer that test here routinely, and we're both initially because we thought we were measuring immune complexes, and then later in a slightly modified way to measure anti-C1q. I think anti-C1q, you know, when you kind of look at the many biomarkers that are used in lupus, anti-C1q still shows up as one of the better ones, which is another way of saying among the many that don't work all that well, C1q is one of the ones that works better than many others. So we stopped offering and promoting the test in part cost effectiveness. This is another marker that's of some value. It is of use, and I didn't talk about it, but the syndrome of hypocomplementemic urticarial vasculitis is associated with autoantibodies to C1q. It's actually the same antibody to collagen-like region. And it, because of how hard it is to diagnose that syndrome and lack of other markers, I think there still is a role for anti-C1q in that setting. There are laboratories that still run the anti-C1q, and there are various approaches to modify the assay so it actually doesn't detect immune complexes, but measures autoantibodies to C1q. I won't go into the technical details. Some of those are better than others in terms of whether that actually works. But C1q binding with a solid phase assay that measures anti-C1q or immune complexes still actually is it is actually a good test for lupus and active lupus nephritis, fluctuates with disease activity and so on. Is it sufficiently valuable for people to order it routinely? And I would say probably not cost effective in that regard. Oh, and Sarah has a great question also. Why do patients differ? Why one organ and not another? And you know, I think it may be that some of Dr. Manick's data would help inform that, and you know, some of it might relate to local charges on structures. For example, that decoration of the basal membrane in the kidneys makes that a high risk organ. The filtration role there, actually, as Dr. Simkin has pointed out, the slit pore in the kidney and the slit pore in the synovium or endothelium are sort of similar in terms of potentially allowing things to leak across. So that might make a difference. For cryoglobulins, of course, temperature makes a role. Lower extremity skin is colder than other parts of the body. Sometimes you see it in the ears like one does for urate tophi deposits. So temperature makes a difference, not explored in these experiments. I'm sorry, this is hand waving because a lot of this is a good question that's not fully understood.
And Dr. Wener, it looks like Dr. Starkeybaum raised his hand, so I'm going to promote him to be able to talk here. Dr. Starkeybaum, did you want to say something?
That was a great talk, and I just want to relate how I met Mart and how he attracted me to the university. So I had gotten drafted after my internship, and I was in Mexico at a conference that he and Ralph Williams were holding, talking about what was active in rheumatology in those days. And Ralph Williams was one of his colleagues at the Rockefeller, along with Chuck Christian and Eng Tan and a variety of other luminaries. These guys became lifelong friends and colleagues and influenced rheumatology all across the country. And Mart was actually a very dynamic, enthusiastic speaker. And he was talking about immune complexes and rheumatoid arthritis I think the self-association of rheumatoid factors. And you could just taste the idea that rheumatoid arthritis was an immune complex disease and we were on the verge of solving it. And because of his expertise and magnetism and such a great speaker, I said, well, when I get out of the Army and back into UW as a resident, I'm going to go into rheumatology. And I never wavered in that. And Mart, led a program with very active research in those days. And as you've talked about, Bill Arend and Dick Pope and a number of others have gone on to distinguish themselves. So he contributed, rigor of science and enthusiasm for the science of rheumatology. So thanks for diving into a number of his research activities.
And thank you, Gordon. I agree. I think rigor, enthusiasm, and the booming voice, kind of a little bit of gravelly, and then this accent you could never pin down. He never totally lost his Estonian accent, but it was subtle and it was very personal, but it was incredible. And that voice, when he was giving a talk, was so exciting and inspiring. But if I'm not saying anybody ever screwed up, but if somebody did screw it up, that same voice had different impact, I would say.
He was fearless about setting it straight. The other thing about his speaking, because he never lost his accent, he always had a bit of hesitation, and you didn't know exactly what he was going to say next, and you had to watch every word and listen.
It looks like Dr. Alkan would like to add something as well. Dr. Alkan, you can speak now.
Yeah. No, again, thank you, Mart. It was a fantastic tribute to Mart Manick. Of course, I knew him very well long before I came to Seattle. He was held in great esteem by all the highest minds in rheumatology. His work was, as you say, focused and incisive. And he had qualities that would inspire fear. If he stood up at the ACR and asked a question, the presenter would tremble. But nevertheless, he would be spot on, precise, and make people think harder. So he was a wonderful leader and great inspiration to many of us. Really, it reminds me we should have had a couple of hours to discuss his work and to let people reminisce about Mart Manick. So anyway, but unfortunately we only have an hour, so have to cut it short, but thanks again.
All right, thank you so much, Dr. Wener. I think that wraps things up, and this lecture has been recorded, so we'll make sure to post this on our website as well and send it around to the division. So, Dr. Wener, I think
before we go, Dr. Sasso asks for a quick comment. I wonder if you see that in the chat. Former fellow, former faculty, now with industry.
Dr. Sasso, let me see here.
Well, I just saw that comment. Maybe it's gone.
Yeah, Dr. Sasso, I think if you can raise your hand on here. Oh, I see. Here we go. Wait.
Raise. Okay.
Okay, I think that you are able to speak now.
Okay, good. Thank you. Are you hearing me?
Yes.
Well, thank you very much. Yes, Mark, I wanted to thank you as well for that kind of very nice review and tribute, and just add to other people's comments a kind of recollection that still has life in 2021, I think, for many, many people, which is that Mark's approach as a rigorous scientist and extremely kind of careful, attentive investigator, I think impregnated the thinking of everybody who worked with him, and to the point that I still speak with people, even people who may have only trained with him for a couple of years and I never even thought of as really sort of bench scientists, still refer to Mart as a person who gave them a kind of a disciplined way of thinking that insisted on having controls, that insisted on analyzing the data carefully, and was skeptical of any sort of sloppiness that they saw in others. And so he really has had a lasting legacy that goes way beyond even the kind of core enduring scientific community that he trained.
Thank you, Eric. I echo those thoughts and appreciate your thoughts and Gordon's and Keith's as well. So I think we really appreciate the rigor that he brought to us, the critical thinking, detailed analytical approaches that we carry on. Thank you all to those of you who have attended.
Thank you, everyone.